CAT, we tackle difficult problems and are really focused on innovation. We want to make a difference in how aquaculture is practiced. The world population is expected to reach 9 billion by the mid-century, and we will have to double food production to feed the planet. Many experts believe that fish will be a solution to animal protein production, and in this sense, it is very gratifying to know that the tools and the product that we're developing can have long-term benefit to society, helping feed people while at the same time preserving the environment. The potential of genome editing as a genetic improvement tool is astounding. Genome editing will enable the industry to introduce improvement in a variety of important characteristics, including disease resistance, environmental resilience, growth, and optimize nutritional value.
In aquaculture, wild and farmed animals frequently occupy the same environment. This means that producers have a responsibility to find a balance in growing fish that can meet customer demand while maintaining stewardship for the environment they share. In fish farming, we may want only females because they don't become sexually mature during grow-out, or males because they grow faster and are more feed efficient. Where farmed fish and wild fish share an environment, an ideal solution would be if farmed fish were sterile, preventing farmed fish from establishing invasive populations and interbreeding with the wild fish. Sterility also improves commercial productivity because in many aquatic species, sterile fish direct their nutrition to growth, not sexual development. Sex control and sterility management are already practiced in some farming operations, but the current methods are difficult to scale, can have other detrimental effects on the health and welfare of treated animals, and can create risk for personnel and the environment. If breeders need fertile fish to produce lots of juveniles for stocking farms, and farmers want sterile fish for better productivity and sustainability, it creates a sterility paradox because fish can't be fertile and sterile at the same time. For the past decade, CAT has been looking for a solution to this problem, and now we are excited to announce that we have solved the sterility paradox. At CAT, we have developed a means of successfully producing reproductively sterile, single-sex populations of aquatic animals using a genome editing approach, and we have devised procedures to ensure that these populations can be created at scale in a commercial breeding program. The CAT solution utilizes genome editing to create elite fish, which are reproductively sterile and single-sex by targeting genes which control sexual differentiation and gonadal development. Small, precise deletions are made in the DNA code of the genes of interest to control sex and stop the development of germ cells into eggs or sperm. Genome editing does not require the introduction of new DNA and results in 100% sterility in production animals while maintaining fertility of the broodstock. In addition, this technology produces populations of fish that are single-sex. A genetic solution using genome editing to control sex and sterility offers a welfare-friendly, sustainable approach to the challenge of producing single-sex populations that cannot breed in the wild. Production and biocontainment of elite genetics leads to improved economic development and food production. Our solution exceeds the current industry practice by producing reproductively sterile populations at commercial scale without using physical shock or chemicals. Alternative sterility methods are not 100% effective and often result in undesirable health and welfare issues. To date, our research team has demonstrated that they can mass produce sterile, single-sex populations of tilapia using a technique that will work equally well on other species of finfish, with 100% efficacy. We have filed and submitted patents to protect our approach in all major geographies in which aquaculture is established as an industry. As an organization, we intend to collaborate with aquatic animal breeders to make this technology available around the world. CAT is already liaising with the regulators in key jurisdictions and working to ensure that the food products will be accepted in domestic and export markets.
In our view, The development of a practical solution to the sterility paradox is foundational. The responsible commercial application of genome editing in aquaculture, since it reduces the risk and removes barrier to acceptance by regulatory agencies, producers and consumers. The technology and know-how that CAT has developed is a platform on which to deliver the promise that genome editing makes for creating major improvement in health, welfare, and efficient production of farm fish.